If you're just joining us, our cameras are live this morning for a forum on DNA research. Right now you see Christopher DeMuth with AEI, expected to give the introductory remarks.
Good morning. My name is Chris DeMuth. I'm president of the American Enterprise Institute, and I'm delighted to welcome all of you here this morning for this AEI seminar on the promise of DNA research. About a year ago, AEI sponsored an informal conference among religious leaders, genetic scientists, business executives, and bioethicists to discuss a range of ethical and policy issues that have been created by the forward march of our knowledge of genetics and the biotechnology revolution. It was a most productive gathering, and at it we resolved to bring the discussions to a wider audience through a series of seminars that would explore these issues in greater detail. And this morning's seminar is the first of them. In the meantime, of course, the issues that we were debating a year ago, such as the patenting of genetic information and genomic inventions, issues of genetic privacy and discrimination, have been powerfully augmented and amplified by the announcement in February of the cloning of the sheep Dolly, and the debates over human cloning, and the administration's recent proposal of a legislative ban on human cloning that has ensued. All of these issues will be explored at this seminar series, beginning with three. Today's seminar is devoted to gaining a greater understanding of what it is that genetic scientists have discovered and have in store for us. It will be followed by a seminar next September 4th on risk regulation and responsibility, genetic testing and the use of information, and a final seminar in November on the ethics and political economy of genetic research, and further ones, I suppose, in the next year. Everyone here is, of course, warmly invited to attend the seminars we will be holding in September and November. For this morning's session, I would like to particularly thank the many distinguished scientists from both commercial and academic research and business executives who have taken time to be with us today and educate us. I am especially grateful to my colleague Clarissa Long, who is an Abramson Fellow at AEI, for organizing the conference and attending to all of the details of concepts, from concepts to logistics, in bringing this conference to reality. I'm going to turn the podium over to Clarissa, who will introduce our first speaker and subsequent ones in the course of the day. Clarissa Long.
Thank you, Chris. As you can see, we have a full schedule today. Our first speaker for the day is Dr. William Haseltine. Dr. Haseltine is chairman of the board of directors and chief executive officer of Human Genome Sciences, Incorporated. He holds a doctorate degree from Harvard University in biophysics and has been a professor at the Dana-Farber Cancer Institute of Harvard Medical School and at the Harvard School of Public Health. He has published over 250 scientific articles, is the editor-in-chief of the Journal of AIDS, and has founded 7 companies, each in a different area of medicine, in the past 16 years. Last year, he received the Ernst & Young 1996 Greater Washington Entrepreneur of the Year Award for biotechnology. Dr. Haseltine will be presenting an introduction to the basic science of DNA research. Please join me in welcoming him.
It's a great privilege and pleasure to be here today to introduce this very interesting forum for the American Enterprise Institute. The topic is one of great interest, not only to professionals who are interested in the fundamental aspects of biology and medicine, but also to all of us as citizens who are concerned about our health and our environment. Because just as we look at our beautiful world today, with its blue skies and its green rolling hills, its lush vegetation and its rich variety of life under the sea, we have to come to realize that all of that was created by living forms from what was essentially a barren rock with water on it. How did that marvelous transformation come about? The very air and oxygen we breathe was not in our atmosphere before there was life on Earth. What happened was living forms evolving over time created first a simple chemistry, then a more complex chemistry that has, in the words of the science fiction writers, terraformed this rock to give us what we now live in as a most pleasant environment. That's how powerful the chemistry of life is. It is powerful enough to transform an entire planet from a bare rock to what we see today. What modern biology has done is allowed us to peer very deeply into the nature of living things, to peer beyond what is visible to the naked eye, to peer beyond what is visible to the microscope, and today, with our modern understanding of atomic science and our modern understanding of how living organisms are built, we can at last unite what have been the two great trends of science, the theoretical understanding of what nature is and how it comes to be from the first principles ranging from energy, conservation of mass, the nature of the atom, to what a living organism is. It's been my great privilege as a scientist to be part of the revolution in human understanding that leads to that great fusion. I am trained as a biophysicist. Think about that for a minute and what it means. It means a physicist interested in biology, or a biology interested in physics, a biologist interested in physics, or a fusion of the two. And indeed, we can now begin to understand the living world in terms of the atoms, the chemistry that make up all of what is in this universe, and therefore make up that which is life. That may seem a far cry from creating better means to prevent, to predict, to cure, and to treat diseases, but in fact it is very closely related. Let me give you a story of what we are doing at Human Genome Sciences and how that fits into this emerging picture of what we are. When I was a student, I worked for James Watson, the man who discovered DNA, and Walter Gilbert, the man who learned how to read the text of DNA. At that time, we thought of the living world as relatively simple. There was an information molecule called DNA, which was the storehouse of individual genes. Genes are the irreducible heritable traits. They were originally defined by Mendel and then by others based on observation that the earlier concept that inheritance was a matter of mixing of two fluids, and we still have that in the concept of bloodlines. If you say it runs in the blood, or this is characteristic of a pure blood or that kind of blood, we still have that nomenclature. He actually defined, through very careful observation, the fact that genes didn't behave like fluids. They behaved like two bags of marbles that were actually fundamentally immiscible, but had different traits associated with them. And one of those irreducible heritable traits for height, for color, for whatever it was he was measuring actually didn't mix with something, but could be separated out in subsequent generations. That's what the origin of a gene was. But later, people came to understand what the physical basis of the gene was. That was a theoretical construct. But during the 50s, and with the discovery of DNA, people came to understand that a gene, this heritable trait, had a physical reality. It was a length of a molecule called DNA, which for all practical purposes you could think of as a two-stranded rope. It's a long molecule with two different strands that has the wonderful property that when you separate the two strands, a new and identical strand can be templated on the other to make two essentially identical ropes. Well, what is that rope doing and how does it make a gene? That rope is like a piece of magnetic tape. It contains information. It has chemicals built into it, 4 different chemicals, which actually spell out a code that contains information. And that code, when processed by the living cell, produces from one length, one product, which is a protein. So at the simplest level, what you have and an understanding of genes is one gene is an immiscible trait that is inherited, and at the other, the molecular atomic level resolution, one gene is a length of DNA made of atoms which contains information that when processed by the cell makes one protein. One gene, one protein. Proteins do 3 different things in the body. Proteins build structures, they build the fibers, they build the physical structures of our body. They are the enzymes that transform one chemical to another. The breakfast or lunch that you're now digesting, depends when you listen to this, is now being transformed by enzymes from one chemical form to a different chemical form by those enzymes. And the third function of a gene is a switch. It turns systems on and off. The most familiar switches to most of us are hormones that act throughout our bodies to turn on or off different functions, but we have a lot of different switches. Altogether, we now understand that a human has, and it may seem small or it may seem large depending on your point of view, about 150,000 different pieces, different genes that make up that collection of a human. So that's where we've progressed up to now. And what happened about 5 years ago was a fusion, as very often happens in science, a fusion of a number of different technologies which allowed us to gain a much more rapid and deeper insight into how we are built at the genetic level. We had this theoretical construct of what a gene is, of how it was made, that a gene is a protein and a protein has these properties, but that was mostly achieved by the study of simpler organisms. When it was applied to humans, it was applied one at a time, and when you're trying to find 150,000 things one at a time, and each one takes between, say, 2 and 10 years to find, you can't find very many of them. As often happens in science, 3 different technologies came together to make the science of gene hunting much quicker, more rapid, and indeed almost automatic. And what happened is over the process of about 25 years, people began to learn how to work with genes. At first, it was a rare art form. It was the one I was trained in. Later, it became easier, and over time, people learn more and more tricks, sort of like cooking tricks, of how to work with genes. The second was a related revolution in robotics. We as humans have become more and more facile at replacing what our hands do with robots, whether it's building cars or building computer chips. People realized that in fact most of what biological sciences did was manipulate things with their hands, and built robots to replace that. Combined with the ability to manipulate genes more easily, you now had robots that could do that, and that enormously speeded up the process. A long time ago, we figured out the advantages of having an assembly line. Once you have robots doing a process, it's just simple leap to have assembly lines do the same thing. And so about 5 years ago, it became possible to robotize the discovery of genes and to put that into an assembly-like array. And that's really how our organization was born. Now that creates so much information that you can't even begin to think about it without the aid of powerful computers. And the third thread that allowed the modern revolution in what is called genomics to occur was the miniaturization, the speeding up, and I should say, very frankly, the dramatic decrease in cost of high-speed computers. So those three discoveries, ease of manipulation of genes, laboratory robots, the ability to handle very large amounts of, and sort through genetic data, has allowed the creation of a new view of humanity. We can now view humans, or for that matter, any other living organism, in ways we could only begin to hope for 10 years ago. That is, we can now define what a human being is in terms not just of their organs, the heart, the lung, the blood, the stomach, the skin, not just the tissues that make those up, not just the cells that go into making up those tissues, or even the organelles, but really at that ultimate genetic level, that irreducible level, one gene, one protein, we can now begin to define the normal human in terms of what genes make up what parts. It's a new anatomy, a genetic anatomy of a human being. We can ask questions now. What are that collection of parts which make the brain, which make the pituitary gland, which make the hypothalamus? What are those genes that make the lining of the blood vessels? What are those genes that make the kidney? That's one question. We can begin to ask and answer questions about normal and abnormal structures. What is the difference between a normal breast cell, a breast cell which is defined as cancer in situ, in place, or cancer that's metastatic? What is a collection of parts that go into making up those different kinds of cells? What are the key differences? We can even ask questions about what are all the parts that are expressed in common by cancer cells as compared to normal cells. Not just one kind of cancer cell, but all cancer cells. Because we have now built, through the systematic application of these new technologies, a comprehensive catalog of what is human at the genetic level. Now, you may have noticed in this description of genomics, the science of understanding how we're built from genes, that the only time I mentioned inheritance was when I mentioned Mendel. You do not need to understand inheritance to know that you have a heart or a lung. You do not need genetics to know you have cells. You do not need inherited patterns to know what genes go wrong in what tissues. Genetics is one, but not necessarily even the most powerful tool that medicine has today in understanding our bodies. And so what you are reading about is the discovery of the gene for Alzheimer's, the discovery of the gene for obesity, the discovery of the gene for this cancer or that cancer, is but a tiny slice of what is a much larger revolution in biology, which is a comprehensive cataloging of the genes that make us up, how those genes make up our normal, not our abnormal, our normal organs and tissues, how they function during normal replacement processes, how they go wrong in diseases, most of which are not inherited, what happens when we are damaged and we repair ourselves, and indeed how we build ourselves from a single cell into a complete organism. Well, how do we go from this knowledge, this broad base of knowledge, which is a new type of knowledge, into creation of new means to predict disease, detect disease early, prevent diseases from occurring, treat disease, and ultimately cure disease? And it's a rather straightforward, surprisingly straightforward problem. Suppose I'm interested in the repair of wounds to the skin. How do I approach it with this new technique? I take normal skin and I take damaged skin that's in the process of repairing. I analyze the complete set of genes, which now can be done relatively rapidly, in a matter of a week or two, a complete set of genes that are now being expressed in those two tissues. I enter that into a computer in what is called the relational database, that bits of information. And now I can ask a question. Give me all the genes that are expressed only in the skin which is being repaired. First of all, I can say all skin, repaired or not. Just give me those components that are being produced in skin which is undergoing repair. Don't just give me those components, give me a list, an inventory of what their relative abundance is. What are the most abundant parts? That immediately informs you as to what is going on in that tissue at its most fundamental level. From that, I can sort through using computer means to pick out those that I think are the most relevant and begin, because our process actually isolates those genes in the process of gathering the information. We have this complete set of genes in the freezer. If one looks like it's an important variable, we can begin to work on it. Well, we have done that, and the answer for that problem came out to be very satisfactory. We found something, a gene, a part, which was only present in large abundance in skin that was being repaired. When we studied it, we discovered it was a new hormone that the body makes that acts at short range, that stimulates the skin to grow rapidly and to repair not only the surface of the skin, but the deep underlying layers, and replace that open area or the cut area with a fine layer of skin and a very thick under support. You may have noticed if you have a wound and it's scabbing over, that right at the edges of the scab, the skin is thicker. Well, what this substance does is actually thicken the skin, make a thicker layer than normal, add blood vessels, add connective tissue, and slowly it thins out. So from asking the question to getting a tool to address the question can be a matter of months, not a matter of years or decades, as was the case previously. Let's take the case that I mentioned earlier of breast cancer. We can and have analyzed breast cancer tumors from a number of different situations, and now can give you a catalog of some 300 parts which are only used in breast cancer cells which are metastatic and not in benign or resting breast cancer tumors. That gives you two things. It gives you markers to distinguish tumors that are benign from tumors that are malignant, or it can do that. And it can give you a whole new set of targets. What drug makers are looking for are things that they can target drugs to, knock out the function of, that won't affect normal tissues. And if you now have 300 parts, genetic parts, which are used only in that breast cancer tissue, which are not used in normal breast cancer or anywhere else in the body, because we've taken a survey of the body, you now have in very short order defined a new means to approach that disease. Those are just two of the many, many types of application of this technology. I think that the future for medicine has never been brighter. We have now a much better understanding at this most fundamental level of how we're made. We are beginning, we as a small organization and we as an entire community, because this knowledge is not held private, is basically humans are very good at seeing what works and adapting it, sharing it with a large number of other groups. We as a set of very curious people are now beginning to understand how to apply this technology to a broad range of problems. Certainly, the applications of this technology don't stop with medicine. We have just conducted a similar set of research, that is a definition of an organism. The organism I discussed with at first was human, at the level of anatomy, physiology, pathology. We've done that for human. We've just about finished a similar description of the corn plant. We now have an anatomical description of what corn genes are used to make the stalk, the stem, the root, the seed. We have a description of how that changes over time and with disease, and how that changes as the plant grows from a seed into a mature plant. And of course, that information can be used for a variety of purposes, including making plants that are more adaptable to a wide range of growing conditions, that produce different nutritional properties that may be desirable and that may be resistant to various forms of disease. So this type of technology, this comprehensive view of genes of an organism can be applied to many other situations as well. The final class of situations which I will describe is this technology is powerful enough to define the entire set of genes of a microorganism, say Staphylococcus or Streptococcus, in a matter of weeks. When we set about to analyze the total genetic content of the Staphylococcus organism, it took us no more than about 3 or 4 weeks to get most of the genes isolated, and a few more weeks to finish the job, a couple more months to interpret that amount of information. So that you can, with this technology, gain a very deep and rapid understanding of simpler organisms. As well, that kind of information can be turned to advantage in creating new vaccines to prevent disease and to create what is rapidly becoming a new set of badly needed antibiotics. So the technology, this new technology of gene discovery, has many applications in human health, in agriculture, and the control of infections. With that, I'd like to stop and open the floor to questions. We have, I understand, about 45 minutes for questioning. So the floor is open. Yes, I'll start in the front and work backward.
How successful has the Genome Project or your own company been in identifying new drug targets?
The question is, how effective has the Genome Project or our company been in identifying new drug targets? There are two basic efforts underway to define what it is that were made at the genetic level. One that I just described, which I would say is the Human Genome Sciences approach to understanding genes. It isn't a comprehensive understanding of all that is inherited. It's more understanding how the genes are used to build our organs, tissue, cells, how they change in normal and abnormal physiology. That is a, I'd say, non-genetic, non-inherited approach. The second approach is, let's read the entire genetic text from beginning to end. That's a government-sponsored project. It's called the Human Genome Project. That is a very big technical undertaking done by coordinated laboratories around the world. The answer to your question, how effective have these two approaches been in finding drug targets? I'm pleased to say that the approach that we have taken has led to the discovery of more drug targets than the pharmaceutical industry can presently absorb. And that's a pretty big statement. This very small group of scientists, using a lot of robots, has very quickly, really in the first 2 years, we were working with a large pharmaceutical company, an international company, SmithKline and Beecham. Within the first 2 years, we mutually agreed that we had found more drug targets for them using this method than they could conceivably use in the next 20 years. As a consequence of that, we decided to share this resource with many other companies. So this has been an extremely fruitful way to initiate new drug discovery, to break what had been a logjam of piecemeal, very slow discovery of new opportunities to create drugs. And I think that's been the general experience. We're not the only ones now to use this technique. By contrast, the Human Genome Project is a much longer range, slower moving type of project, and I'd say they've come up with a handful of possible new targets, but not very many. It's been a different approach. Its philosophy has been one more based on science and deep understanding of everything that a human is made of, regardless of whether that codes for or that specifies something that we actually use. Let me be a little bit clearer on that last point, and that is, when I described this rope of DNA, you might have imagined that every bit of it is used. That would be very far from the case. if this were a magnetic tape only that was, say, 100 yards long, maybe only 2 feet would actually encode useful information, and the rest would be spacing. So only about 2% of the entire DNA molecule that's encoded in a human being actually specifies information that goes on to make useful parts. The rest appears to be packaging. So the Human Genome Project is reading the genes along with the packaging. The method that I described just looks at the genes themselves and how they're used. But it's been a very, very fruitful, very satisfyingly fruitful means for finding new drug targets. Yes?
You mentioned that there are more drug targets than can be processed in 20 years. What could be done further down the line in the drug companies or any other way that would shorten this time?
The question is how, if we have more drug targets than we can use now, given our current level of effort, what could we do? I think a number of things are being attempted. Now, the type of systematic use of robots and automation that we have seen and witnessed in the discovery of genes is now being applied to the discovery of chemicals, in the sorting through of those chemicals for medical utility, and also attempts to automate the effects of those chemicals on biological systems. So there is a systematic effort now to catch up to what is leapfrogged ahead of current technologies. That has only, in my opinion, at this point been partially successful. There's some wonderfully innovative ideas that marry computer chips with biotechnology, with living systems, actually integrate them on the same area. And I think they will be successful. So I think in the future we can look forward to really a cornucopia of new ways to prevent and treat, and I believe ultimately cure disease. But we do have a gap at present, as you pointed out.
What is the future of germline research in genomics, and what are the issues, and what is the future?
The question is, what is the future of germline research? For those of you who don't think about germline research every day, germline research means changing in a permanent way the inherited content of an organism. The current state is that it's certainly possible to do with bacteria, with plants, and with many animals. Certainly with laboratory animals, the inherited capacity of those animals is manipulated and altered for experimental and medical purposes on a routine basis daily throughout the world. It can be done. It is also being done for agricultural purposes, certainly with plants and with some food animals. There, to my knowledge, it could be, but it has not been done with human beings. And there is a major debate underway, which I think will intensify over time, as to whether it should be done. Yes.
As you're aware, I think beginning with biotechnology and increased by genomic research, we've seen an increasing trend for scientists such as you, who used to spend all or most of your career in the academy as rather pure academicians, to take your discoveries and your minds into profit-making enterprises and into businesses. You're aware that there are some people who are made rather nervous by this, that we're losing a cadre of scientists who are responsible only to pure discovery, so to say, and losing them to some degree to secrecy and a vested interest. And others are very excited about this, thinking it'll simply promote faster research, as capitalism might want to do. Would you just comment on what you've observed about the strengths and the weaknesses of this trend, and assuming that I've correctly identified what is an increasing trend?
The question is there an increasing trend for scientists who are in academia conducting pure research to move into the area of more applied research, and is that a loss for our community, and does it have some attendant risks? I think we have a very healthy scientific community in academia, and we are building a very healthy community of people who are applying the knowledge that is created in both areas. So I don't think the basis of the question is founded. I don't think we're at a risk at this point of losing fundamental research. It's very healthy. The budgets continue to be strong, and we have and continue to train a large number of people, and there are a very healthy group of people in academia. I think it's very important, however, to keep that. We must continue to train manpower here in this country, and we must continue to support fundamental research if we are to realize the promise of this new technology. On the other hand, I don't think there has ever been anything that is really pure research, and it's not so easy to separate research from development. Let me give you a very specific example from my own career. I've always tried to do two things in my career: create new knowledge and apply that knowledge to medicine. So when I was at Harvard at the Dana-Farber Cancer Institute and I was working on the AIDS virus, I had a very explicit goal in mind: to understand that virus and its working parts and to reduce it to a form that the pharmaceutical industry could work on to find drugs. I saw that as my mission as an academician to do. So my work was not to understand the AIDS virus as a work of nature, but to reduce knowledge of the AIDS virus to a form that the pharmaceutical companies could work on. Because I knew, as most people do, that academicians and the NIH don't produce drugs. They don't develop cures. It is the pharmaceutical industry that actually is responsible for improvements in health based on knowledge created. And it's not even the case today that new knowledge is not created in the pharmaceutical and biotechnology industry. I would say if you look at the journals today, there is an increasing similarity between the work which is published by academicians and the work that's published by pharmaceutical scientists and by biotechnologists. So it's mixed. Do I think there's a danger of it going too far? No, I think it's a very healthy interaction. And we see an exchange of personnel from people going from academia to the pharmaceutical and biotechnology industries. And now that those industries have been well established, we see a reverse flow as well. So we see people who've made careers in the biotech industry going back into academia. There's a close interface between the two enterprises. And I think in this country we've got it just about right. The door should be open both ways. Yes?
Thanks. I was struck that you are by training a biophysicist, and this area is often spoken of as an area of biochemistry. Two questions. First of all, does it make any difference anymore whether one is coming from a biophysics background or a biochemist background? And secondly, do you have any comments about the fate of biophysics as training? It seems to be put on the side of the training of doctors and of other people in this country, at least.
The question has to do with the relative merits of biochemistry and biophysics, and that's an esoteric question. In my own case, I felt that a very deep grounding in the fundamentals of nature, which you get really through a study of physics, was essential for a career in medical science. I found that it was extremely useful to have a set of tools for approaching problems that were really unconstrained. I was trained as a chemist as an undergraduate, then had a physics training as a graduate student before becoming a biologist. And I did that very deliberately so that I felt comfortable with a full range of tools, so I could define the problem and then find the tools to solve it, rather than have the tools define the problem. And I think for that reason, it's really important to have people exchange fields. It's important for biologists to become computer scientists which they are today, and it's important for physicists to become biologists and chemists to become computer scientists and biologists. I think there's a wonderful area of interchange now, and some of the most exciting developments and approaches to the kinds of problems that were referred to earlier, which is how do you translate this enormous increase in genetic knowledge to new products, will come from the fusion of the semiconductor material scientists with biological impulse. So that kind of fusion, I think, is very healthy and fortunately is going on today. There's a whole new field that has sprung up called bioinformatics, the use of computers and computer technology to understand biological phenomenon. If you look through the scientific literature today, every journal is filled with ads for bioinformaticians. It's a new field and one which I would encourage any young person to think seriously about because it's a huge void today. It's a field that didn't exist 5 years ago or existed only in a very simple form 5 years ago. So I think that all these trainings are very important. We should train people broadly in the sciences. Yes.
Now that you are in a for-profit mode at HGS, given the new debate on private and public databases, how do you as a company decide what's going to go where? When do you publish patent issues? There's the report of the new asthma genes that have been discovered but not yet published, but certainly press released. And as a sort of a corporate philosophy, how do you decide what's going to go where?
The question has to do with, as a corporate entity, how do we handle intellectual property, when do we decide to publish, and how we decide, in your words, what goes where. The answer is surprisingly similar to what I did as a professor. And that is, what is obliged for scientists working at the NIH or any government facility to do. And that is, first patent, then publish if you think it has a use. When I was a professor working on the AIDS virus, we would find what we thought was a new and important drug target that we could reduce to some useful form. My institution, Harvard Medical School and Dana-Farber Cancer Institute, required me to give them that information, allow them sufficient time to file a patent before I published it. That is almost universally true throughout academia today. And I think people might find that surprising. At the NIH, it's also true that if you make a discovery, you are operating under the terms of the Bayh-Dole Act. The Bayh-Dole Act specifies that any federal scientist or federally supported scientist who makes a discovery has a positive obligation, that is a practical application, to make sure that intellectual property is protected so the government can then transfer that to the private sector for its proper exploitation in commerce. So that is a positive obligation which all scientists working for the government or supported by the government work under. And so what we do at our company is basically similar. If we have a practical application which we've reduced to practice and we think we can teach others, we file a patent application. After that, we feel confident that it can be released. There is another type of information, however, which industry generally withholds, and we follow that practice, and that is if it is new and useful knowledge, but has not yet been reduced to the point where it is practical and can be taught to others, you then hold that private. Let me give you two examples. One is the chemical, say the pharmaceutical industry, that creates a wide variety of chemicals. They do not reveal the structure of all those chemicals. It is only when one of those turns out to have a practical use that is disclosed in a patent application and therefore is made public. Patent applications are, by their nature, public disclosures which are intended to teach others how to do the same thing. Similarly, with a lot of genetic information we find that we don't have yet a practical application for, we do not disclose that, nor do other companies working in this area. Let me extend a little bit beyond the question to a problem that I perceive. There is a difference between one type of genetic information we find, which we don't have a real use for, and the kind that we do have a use for. And I think there is a danger in releasing too much information first. Let's go back to the pharmaceutical company with its broad library of chemicals. If it early on revealed all those chemical structures, it would destroy the subsequent ability to provide patent protection for those chemicals. Because once you make it public, it is no longer new. And if it's no longer new and unknown, you can't protect it. The same is true for genes. If you were to publish a text of all genes, you would undermine the future ability of someone who wished to make a medicine from that gene from doing that. So I believe, I have the personal conviction, that much of the future of medicine will be the use of genes themselves and their proteins as medicine. I referred very briefly at the beginning to something I called restorative medicine, and I gave you the example of the natural human substance to repair skin. Well, how do you make a product from that? You take the gene, you produce the protein, which is the hormone that heals the skin, and you claim the gene and its protein in a patent application. You can do that and be encouraged to invest if you have the rights. However, if someone else had published the text of that gene, I could not claim that drug uniquely and would be open to competition. So, for example, I might have taught somebody how to use it for the skin, but it might also be useful for repairing of the intestinal tract. Somebody would know how to make it, use it to repair the intestinal tract, sell the drug, and the user might use it for the skin or the intestinal tract. So it's a very serious problem, and I think one that is not adequately addressed today. And those who think you should throw open the chest that contains all the genetic information have not thought through, I think, very carefully the problems that that would mean for a subsequent creation of what I believe will be a new and better type of medicine. So that's impact, and then we'll go to the front again.
I will speak it more in economic way, okay? Economically, do you think the drugs prices will go down once these genome studies will have remedies for the various illnesses we have now? And do you think new forms of illnesses, like AIDS, for instance, which came in recently, will arise once these studies are in public use?
The question was, will drug prices come down as a result of these technologies? And if you do cure a variety of diseases, will new ones appear? Let me address that question a different way. You can't, the pricing of drugs is really a matter of the marketplace. It's a question of supply and demand. And so I can't predict what the individual prices of drugs will be. I can say, however, that I think that the genomic science can make a major contribution to reducing overall healthcare costs. I see a time when medicine will be based more on the prediction and early detection and prevention of disease than it is on intervention. I see moving from a interventive paradigm to a preventive paradigm that will be based on thorough knowledge of our genes, early detection, and a wide variety of new techniques to prevent and to restore to normal function that which could go wrong, has gone wrong, and has already happened and damage that is done. That will have the overall effect, I believe, of substantially reducing the overall healthcare budget, which is heavily weighted toward intervention and to treatment of people after they have become ill. I see a change in the overall, a potential change in the overall allocation of healthcare resources. While the overall cost per person may shrink, the proportion which is in the pharmaceutical sector, prediction, detection, prevention, treatment, and cure as increasing per person. So the answer to your question is a little bit more complex than will one drug go up or another down. Now the next question you asked was, will new diseases come about? And I'm afraid that we will be prey to a number of types of new diseases. I think that just as we've seen a new, not a new virus, but a new form of an old virus sweep through our populations, worldwide populations, that is the AIDS virus, I fear that we will have other such infections from the non-human world. We will certainly have a variety of new bacterial infections that are resistant to the current generations of antibiotics, and there will be a never-ending war of humans, or a never-ending accommodation between humans with a great biomass that surrounds us and supports us, I should say. It's not our enemy, it's our friend, but it also has its tendencies to invade our bodies and use us as part of its environment. So we are going to be in a continual non-stable equilibrium with that, and must continue to find new ways to fight that. I think there will be very deadly worldwide viruses that sweep our populations, such as the flu epidemic of the World War I time. And I think that we are long overdue for such a new epidemic. There are things we can do to help combat that. And then in maybe the same semi-humorous tone that you asked your question, I will respond that because we're living longer, there are diseases which we define as diseases which we never defined before. So before, let's say, even 50 years ago, 20 years ago, we defined old age as old age. People died of old age. We don't now think of that. We're now providing a new construction of aging. Aging is now an assembly of ailments. It is Alzheimer's, it is atherosclerosis, it is kidney failure, it is high cholesterol, it is a variety of cancers, it is thinning of the skin, it is erosion of the vascular system. We now define that process as a series of diseases which we did not define as diseases heretofore. And it has a profound consequence, because when you define something as a disease, you approach it with a treatment or ultimately a cure. And I think that's very positive. So we are, as we are extending the boundaries of age, we are defining new diseases which are not extrinsic but are intrinsic, and at the same time we're beginning to devise means to counteract what would have been called the ravages of age.
I'm going to ask you to put on your soothsayer hat. Looking out 30 years, what can we expect medicine to be like, and how will it affect our health and lifespan, looking specifically at antibiotics and lifestyle diseases like cancer and cardiovascular disease? And I won't hold you to it.
30 years from now, we'll be back here discussing it, I hope. I see the potential of a much better type of medicine. I referred earlier to a switch from interventive to a preventive paradigm, but preventive not so much in the sense of clean water and vaccinations, which we now term preventive health, but preventive health in terms of a very accurate prediction of what is likely to occur in an individual's lifetime with predisposition to disease. I see a series of means to detect what is going wrong early, and a new series of gene-based approaches to preventing those things which could happen from happening. And then, if something does happen, a series of gene-based interventions to restore to normal, full health that which has gone wrong. Now, there are a variety of different ways that will happen, but I think that is a very happy future for medicine. But as all happy futures, it doesn't have to come true. I see as many barriers as I see opportunities. I see barriers that can be solved by some types of legislation, such as genetic privacy and discrimination. As long as we have the specter that if you have a pre-existing genetic condition, you cannot receive health insurance, we will have a serious problem. As long as we don't reimburse, which we do not now reimburse, for genetic tests or other types of predictive tests that don't have a cost benefit within 3 years, we will not have those tests because no one or very few people will pay for them. I see a need to train and retrain our people and our healthcare services to function more in a preventive than an interventive mode. And there are a number of barriers to realization. I've called for in the past a series of commissions and studies and groups like this to focus on what will be barriers to a new preventive health. And I think we need an integrated, systematic approach rather than a piecemeal approach to those problems, because there can be a very bright future and people can live much longer, healthier lives. But all that's good need not happen, and I see many barriers to that very happy future.
What about you talked about the relationship between genes and physiological processes. What about the relationship between genes and mental processes, the brain, mental diseases, language, these kinds of things? Are these links being explored, and has anything been discovered?
Yes, the question is, we talked about the links between genes and physiological processes such as heart disease, kidney disease, and wound repair. Are there similar links between genes and mental processes, both normal and abnormal? And the answer is yes, they are being systematically explored. We have now a very clear genetic picture of how the brain is built and how the subcomponents are built, and in fact an emerging picture of what happens in health and disease in the brain. What is emerging is a picture of human complexity far beyond what was imagined just a few years ago. Let me give you one or two small examples, and that is, let's take a gene which most people, or at least the effects of which most people are familiar with, the serotonin reuptake protein. Basically, it's the target for Prozac and for Paxil and other such antidepressant drugs. One way of thinking about it is, here's a gene which may exist in different levels in different people, and if you have too much of it, it's too efficient. You have low levels of serotonin. If you have low levels of serotonin in the space between the nerves, then they don't function as well and you feel lousy, you feel depressed. You plug that little vacuum cleaner up, you have more serotonin, you feel better. Well, that's one view of it, but what we're now realizing is different people not only have different slight mutations in that gene, but they're of different fundamental sizes. It's as if you cut the gene and then tailored it to the individual person. We're finding that to be a widespread phenomenon throughout the brain. We find it in the Center for Appetite Control, that it's not just inherited changes in the text, it's actually how that gene is cut and pasted together differs from individual to individual. So we're learning a lot more about individuality, what determines some of our really individual traits. And I think that that kind of knowledge will be very important in determining what we can do about some of the abnormal mental states that people experience, whether it's schizophrenia, depression, euphoria, etc. And I think will form the basis, and is already, I should say, forming the basis for a new kind of neural physiology and neurochemistry, which will be manifest in a series of new drugs. So there's great progress which is being made. Yes?
Who is financing academic research, particularly pure research, and to what extent are there grants dependent, or the definition of grant require some suggestion of a practical application?
Right. The question is, who's funding academic research? Let me first take exception to a word that's been used repeatedly, and that is academic research is pure and industrial research is not. I do not believe that. I don't believe in that distinction. And academics aren't any purer than anyone else. And industry is no more corrupt than anyone else. I believe that there is fundamental and very important research being done in both. Who is funding the academic research? Well, it's a mixture, actually. When I was a student, it was about 90% of the federal, actually there's been a history in the U.S. Before World War II, it was about 80% private foundations. After World War II, it was about 90% federal government. Today, at any successful medical school or university, it's about 70% government funding and 30% private funding. That may be even shifting a bit more to 65% government, 35% industrial research or industrial supported research. I believe that there are important distinctions to be made between the two and important separations that must be maintained if you're doing research in the academy that's supported by federal or by industrial dollars. But you have another question behind that, other just than the sheer numbers?
No, I thought the answer would be much as you gave it, and I really wasn't concerned about, as you picked up, purity.
Right.
But I was wondering to what extent does anybody engage just purely not with a target problem as you did with AIDS research,
right,
but whether they're just looking at aspects of just purely the genome structure and trying to find out more about where things work.
Yeah, there is fundamental research being done on those organisms, but I should say that I've looked at that problem actually as an academic for a long time. I've been very interested in the problems of who supports scientific research for what purpose, not just in our time, but in all times. And research is always funded for a purpose, whether it's a space program, whether it's material sciences, there is always, and I think there always should be, a societal purpose. Whether it's an immediate application, I think that's where the boundary should be drawn. I think there is room for substantial support of fundamental inquiry in certain directions without any immediate practical benefit, just as there must be practical benefits for some other types of research. Yes.
As a proponent and developer of genomics, you've presented us with an account of the promise. The public sees in all of this something of a peril. Are there any perils? Should anybody be worried about anything here? Could you put on that hat and comment?
I think there are obviously concerns, as there have always been concerns about new technologies. I think understandably focus on the promise, and I think that's appropriate. I don't think that the perils are as dire as people think. Most people who are working in this field do so with a clear intention of improving the lot of mankind, whether it's by improving our health or creating better sources and improved sources of food or other materials even for our use. I think where I see some perils are actually in the interface with how this knowledge gets used. The most immediate problem we have is a problem of information about individual health destiny. We now are gaining an increasing sophistication to be able to predict what an individual's life destiny would be in the absence of intervention. It, of course, assumes that there won't be an effective intervention. And that kind of knowledge should be protected. Individuals must be, in my view, protected against the adverse consequences of dissemination of that information. Job discrimination, insurance discrimination. Because that information is coming. And I think that's probably the area that I see as the clearest and most present problem. I see the questions of fear that we're going to create new races of monsters is very remote and extremely unlikely.
Thank you, Dr. Haseltine. And ending on that note, I'd like to take a break for just a few minutes before I introduce our next speaker. And let's hear it for Dr. Haseltine.
After a short break, we'll return to this forum on DNA research hosted by the American Enterprise Institute. When we return, Margaret Liu from the Merck Company will discuss drug-resistant infectious diseases. Right now, we'll take a look at the Library of Congress and get an overview of last week on Capitol Hill and find out what's coming up when Congress returns.